Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and a very good day. So welcome back to our second part of the session three. So today we're going to look at two very important things. Number one is cell wall, okay, and number two is protein synthesis. I know some of the content that's going to be presented is kind of like similar to what we have learned previously. So hopefully we can carry on with the topic that I'm going to deliver, as well as learn a thing or two from this particular section of the lecture. All right, so as we all know, bacteria has generally two different types of cell wall. Number one is category of what we call as gram positive. Number two is category of gram negative. And both of them are generally bacteria. And while we are focusing on cell wall, we're going to look at it after these few slides. But just on the recap on what is gram positive and what is gram negative, gram positive is when you have a peptidoglycan, which is a cell wall layer, periplasmic space, and plasma membrane. So you only have a three layer on your outer surface of the bacteria. However, for gram negative, you start off with you have an outer membrane, okay, peptidoglycan, which is a cell wall, periplasmic space, and thereafter you have your plasma membrane. So there's four different types of layers. What we're going to focus on is just looking at the cell wall. Okay, so this is reference number 7. You can read through a bit about cell wall, the discovery, and whatnot. So generally, cell wall is a tough and rigid mesh of a networking structure made up of peptidoglycan. So that's why you can see over here it says peptidoglycan. Okay, so what is peptidoglycan? Peptido, for those who have perhaps noticed, peptido means peptide, glycans mean sugar. Okay, so it is made up of a polymer of peptide and sugar. Okay, and cell wall are generally highly conservative, regardless of what type of bacteria it is, whether it's gram positive or negative. So it's conserved in a sense that the building block of the cell wall is more or less the same. Okay, and it is also a very polymeric structure in the sense that when you're talking about polymer, normally when we think about polymer, especially in your polymer class, is when you're thinking about polymer as a straight chain. Okay. However, for a cell wall, the polymeric nature of the cell wall is not just two dimension, but rather a three-dimension polymeric space. And if you imagine that you have like a bulk of a linear polypeptide chain, it goes up, down, left, and right, and they are all connected or networked, linked to one another. Okay, so this particular cell wall is normally is an alternating beta 1,4 link, and ester glucosamine or NAG, and an acetylmuramic acid NAM. Okay, so it's an alternating beta 1,4 link between NAG and NAM. Okay, so this is an example. So you have NAG over there. So the way uh, identify whether it's NAG or NAM is just looking at this glucosamine, while the another one is NAM, okay, muric acid. And some of them are generally built by these two main enzymes within the bacteria. Okay, number one we call it as penicillin binding protein or enzyme. Another one is glucotransferase enzyme. Okay, and these two enzymes are similar in the sense that the function are similar. Some bacteria have a different structure. Some bacteria have a combined PBP and GT, while others have separate ones. But nonetheless, the function of these two enzymes, whether it may be a single or a double enzymes, are similar. Okay, so this is the structure. Just again, I think this is a recap on the structure of a cell wall. For gram positive, what you'll get is you get a very thick layer of cell wall. Okay, while for gram negative, normally you have just a very thin layer of cell wall. And how does it works? How does cell wall being built? Okay. So for PBP, penicillin binding protein, it's a transpeptidase. It forms bond between the peptide side chain, forming a cross link, while glucotransferase is an enzyme that link between like them. So one forms the bond between peptide side chain. You just focus on peptide side chain, while the other one is the ones that link between alternating monomer. Okay, so this is what I mean. Some bacteria contains high molecular weight of PVP, a penicillin protein, but the function of PVP and GT are both. Okay, and if you look at the image over here on the right hand side, so this is generally how each peptidoglycan is linked. Okay, so imagine that this purple, pink purple is your neck and dam, and then you have a side chain from the neck and dam over here. So if you have one polymer down up here and one polymer down here, your what PPP does is basically cross-linking the two peptide chains. Okay, so it's cross-linking on here. And finally, when it PVP leaves, you get a network structure of your penicillin binding protein. So I think I forgot to mention a little bit down here that the cross-linking um, structure on neck and them polymer is basically an L-ala D-gluco L-lysine D-ala tetrapeptide. Okay, so linking does is basically linking D-ala D-ala. Okay, so D-ala from one to D-ala to another peptide chain. Okay, so what's important on this particular slide is for you to recall a little bit on what we I'm gonna talk about, which is protein uh, biosynthesis. Okay, so for cell biosynthesis, that's it. I'm gonna look at it again when we cover on the prospect of how antibiotics inhibit cell wall synthesis. But generally, that's it. This is the recap on what is cell wall synthesis. Okay, so membrane structure again, you have a lot of proteins on your membrane, and generally, each one of these protein can be a target of a drug design and synthesis. All right, let's move on to proteins biosynthesis. I'm not sure why my picture is big over here. Just going to look at very two simple things, which is function and inhibition of protein cell wall synthesis. Now, interesting thing about protein biosynthesis is that the research on it has been going on way back a long, long time ago. Okay, so 1950s. So and it is now known that protein biosynthesis generally. Okay, so I'm talking about it generally because it depends on what type of molecules, what type of organism we are talking about. There could be a few stages, but in the general principle, it's a two-stage process. Number one, transcription. Number two is translation. Again, I'm sure this is a revision for you guys, but nonetheless, I just want to recap again so that we can actually look at how these are related to antibiotic synthesis. So transcription is a process by which the mRNA is synthesized from DNA. Okay, so you have DNA to mRNA. Okay, that's messenger RNA carrying the information from the DNA. And then afterwards, you have the translation where the enzyme or the process itself decodes the information of RNA into an ordered arrangement of amino acids. So this is where the real protein synthesis is taking place. And this translation process happens on a protein complex called ribosomes. We call it a protein complex, not simply just a protein, because as you will see after this, ribosomes are not just made up of 100% amino acid or proteins. Okay? So it's a complex made up of rRNA, so ribosomal RNA, plus some peptides, so to say. Okay, so make up all this particular complex. And how, just to show the significance of this particular complex called ribosome, is that back in 2009, Thomas Sint, a Sterling professor of molecular physics and biochemistry, was awarded a Nobel Prize. Okay, so Nobel Prize is one of the prestigious award in science, and this particular professor received this award in chemistry for the study of structure and function of ribosome. Okay, so it's studying the structure and function of ribosome, and in 2009, together with Thomas Venkatraman from the MCR Laboratory of Molecular Biology and Adayona from the Wisma Institute of Science. The three of them actually got the same Nobel Prize, okay, in looking at structure and function, so to say, of ribosome. Okay, so from the work of these three independent researchers from three independent institute, of course, they are not the only one that is doing the research. Normally, when you do something like a big project like this, you do have your students, you have your postdocs, postdoctoral, you have a team generally, but you are the head of the particular research laboratory, and these people managed to get Nobel Prize. And I was blessed with the opportunity to actually meet some of the Nobel laureate, including Ada Yonath. You can see down here, that's me. Okay, back in 2010 or 2018, sorry, in Japan, where Ada actually mentioned about the research that she completed, particularly focusing on trying to get the structure of ribosome. And she mentioned that her work took roughly about between, if I'm not mistaken, about 10 to 15 years to actually be able to visualize this is what ribosome is. And then together with Thomas, managed to kind of like figure out what would be the function of this particular ribosome. I also met a few other Nobel laureates, as I mentioned over here. Professor Kajita, Stoddard, Sharpless, Menotz, Kobayashi, and Hunt. So these are all the bright people talking about the research that they've done that generally bring a very, very positive impact to human life, so to say. Okay. All right. So again, let's move back to ribosomes. So ribosomes are made up of protein for stability and ribosomal RNA or rRNA, as I mentioned just now. Okay. And RNA is basically on the catalytic activity. So this is where the catalytic of protein synthesis takes place. And I know, I'm sure, at least I recall, not because I'm teaching this, but because I've learned about it back when I was doing my degree. It's the process of protein synthesis, which we're going to look at in a few slides. Okay, so ribosome contains two main subunits. So one is one large subunit, another one is a smaller subunit. The smaller subunit contains the mRNA binding site. So this is the RNA from the transcription comes in, and the larger subunit contains tRNA binding sites. Okay, three tRNA binding sites. tRNA is, again, for those who can't recall, so tRNA means transfer RNA. So that RNA function to transfer amino acid into ribosome so that the polymerization of amino acid or protein synthesis can take place. The three sites is called as A, P, and E. So A is amino acid, B is peptidyl, and E is exit site. So one is amino acid coming in, amino acid coming in. P is when the polymerization actually happens, and then once the polymerization happens, there will be an exit site for the tRNA to move out and being recycled again to bring another amino acid in. So, in principle, ribosomes can be found either freely floating in the cytosol or bound to endoplasmic reticulum, especially in eukaryotes like us, humans. Okay? And ribosomes differ in size in prokaryotes and eukaryotes. Okay, so in prokaryotes, it's called as 70S, while eukaryotes it's called as 80S. Now, this is just a link for you to view on your own free time to see what does this 80S and uh, 70S means. And this is not really a mathematical 70 and 80S, where if you look at down here, 70S is made up of 50S plus 30S. So it doesn't mean that 50 plus 30 equals to 80, but rather in this particular science, 50S plus 30S equals to 70S. Okay, so this is one of the mathematical jokes that you can actually make to your friends. And S over here, it's not an unknown or algebra, but rather a survey unit. So it's a unit. So 70S is a unit. It's not really a mathematical per se. And S over here, if you look at the YouTube video, is talking about sedimentation. So this is where a lot of time ago, where the technology is not that advanced, scientists when they try to classify proteins, they use this particular unit. So again, this is a structure of ribosomes. If you look at over here, this is a very, very complex, and I'm just going to the right hand side very quickly. So gray and orange lines. So this gray and orange line are structural RNA, meaning that they don't involve in the reaction per se and on the catalytic activity, but rather trying to make sure that ribosome stays as a ribosome, while the RNA in ball and sticks over here is when the catalytic RNA, rRNA is. Of course, you can always go back to protein data bank, search for ribosomes, and you can see the structure in 3 dimension, and you can move it around and see how it's actually look like. So let's move on a bit more detail on the transcription process where you actually transform DNA to RNA. Now, if you think about it logically, we only have one set of our genetic material, right? So we only have one type of DNA. And we've also learned that this DNA, at least in humans, is complex into 23 pairs. And therefore, if you're thinking about it, so and only each pair is more dominant. One is recessive, one is more active, and one is passive. If you think about it, if you only have one DNA, how can you produce a lot of proteins, right? Because if one DNA only produce one DNA produce only one RNA, and therefore one RNA can only produce one protein. So it will take time to actually produce a lot of proteins, especially as we all know, our body produces protein in a very, very quick and large scale. So what really happens is basically, even though if it's one DNA, so you can see over here 3 prime and 5 prime, and so this is indicator of a DNA, and RNA polymerase is producing mRNA from this particular DNA. Okay? So this is not DNA, so this one should be protein, right? So what should actually happen is as the mRNA being produced from the DNA, from the transcription, translation process are actually happening very, very quickly, whereby it's not just a single ribosomes attached to RNA and produce one protein, but rather you can have multiple ribosomes attaching to the same RNA, and then the process kind of like occurs simultaneously as depicted in this particular picture. So even though you have one DNA, so that's your DNA, the red ones over here is your mRNA. And what happened is you can have multiple types of ribosomes producing proteins at different levels. So because since the direction of translation is going that way, so this one is normally will have, say for example, for the sake of argument, one amino acid, that one is two amino acid, 3, 4, 5, 6, 7, 8, and so on and so forth. Okay? As you move along the direction of translation, you get more and more complete protein synthesis. Okay, so this is how from one mRNA you can actually produce thousands or even millions of uh, proteins that serve as a certain function inside our body or our cell. So this is just looking at the amino acid, peptidyl, and exit. So the three sites. Okay, so you can read it on your own. Again, this is just a refresher. We've learned about this in high school. Okay, so I'm just gonna touch about it because these are the processes that is involved in protein biosynthesis. Okay, so on the quick one, we've looked at antibiotics generally. On the part one, part two, we've looked at mostly on the target, which is generally looking at cell wall and protein biosynthesis. We've already looked at bacteria and animal cells in the part one section. So this is the end of a full part one, so to say. Okay, but I'm going to call it part two. And we're going to look at specifically more on how inhibition of this particular two targets, cell wall and protein synthesis, is actually useful for antibiotic production. So that's all from me. Go and watch part 3.